The next metabolic state is the postabsorptive state, or the fasting state. This is where the GI tract is empty and energy sources are supplied by the breakdown of our body's reserve. The catabolism of fat, glycogen, and proteins exceed anabolism. The goal of the postabsorptive state is to maintain blood glucose levels between meals by making glucose available to the blood and by promoting the use of fats for energy. Glucose sparing saves glucose for organs that need it most, such as the brain. So, with the goal to maintain blood glucose levels during postabsorptive state, what are the sources of this glucose? The first place our body goes for glucose is the liver, where glycogenolysis breaks down glycogen to procure glucose. When the glucose from the liver is exhausted, glycogenolysis in the skeletal muscles releases glucose from the stored glycogen. The next way our body procures glucose is through lipolysis in adipose tissues in the liver. Glycerol is then used for gluconeogenesis in the liver. Lastly, our bodies can break down cellular proteins. This is the major source of glucose during prolonged fasting. There is a limited amount of protein that can be broken down before cell damage results. The amount of body fat determines how long a person can survive without food. During prolonged periods of fasting, our body uses more non-carbohydrate sources to conserve glucose. This process is termed glucose sparing. More fat and protein are converted to intermediates than can enter the citric acid cycle. The brain uses the bulk of glucose while other body cells switch to fatty acids as a fuel source. This results in the production of ketone bodies. After 4 to 5 days without food, our brain will start to also use ketone bodies as a fuel source. Figure 24.22a illustrates the major events and principal metabolic pathways of the postabsorptive state. The major metabolic thrust is catabolism and the replacement of fuels in the blood. Proteins supply amino acids, glycogen supplies glucose, and triglycerides supply glycerol and fatty acids. The major energy fuels during the postabsorptive state are glucose through glycogenolysis and gluconeogenesis, fatty acids, and ketones. Glucose, fatty acids, and ketones go through cellular respiration to produce carbon dioxide, water, and ATP. During the postabsorptive state, the liver converts amino acids to keto acids and then to glucose. Figure 24.22b further illustrates the postabsorptive state. Our first source of glucose is stored glycogen in the liver. Next, we can also procure glucose through the stored glycogen in skeletal muscles, which is converted to pyruvic acids and lactic acids through glycolysis. The products move to the liver, and they are converted to glucose. The third source is triglycerides in adipose tissue, and the liver, in adipose tissue and the liver. They are converted to glycerol and fatty acids. Fatty acids are converted to ketone bodies, which can be converted to ATP. The last place our body looks for glucose is skeletal muscle proteins, which can be broken into amino acids, then in the liver are converted to keto acids, which can be converted to glucose. In the postabsorptive state, the sympathetic nervous system interacts with several hormones to control the events. It is more complex than the absorptive state, which only uses one hormone, insulin. The postabsorptive state is triggered by reduced insulin release as blood glucose levels drop. Glucagon is our hyperglycemic hormone, meaning it increases our blood glucose levels. Its release is stimulated by declining blood glucose levels and rising amino acid levels. Glucagon promotes glycogenolysis and gluconeogenesis in the liver, lipolysis in adipose tissue, resulting in fatty acids and glycerol being released, and the modulation of insulin's effects after a high-protein, low-carbohydrate meal. Insulin responds to high amino acid blood levels. If a high-protein meal is consumed, hypoglycemic conditions may result, and that is not good for the brain. Glucagon modulates this, ensuring adequate blood glucose. This figure illustrates how glucagon stimulates a rise in blood glucose levels. Lowering plasma glucose and rising amino acids stimulate the alpha cells of the pancreas to release glucagon. Glucagon stimulates glycogenolysis and the gluconeogenesis in the liver. This increases blood glucose levels. It also stimulates the breakdown of fat in adipose tissues, which increase plasma fatty acids. Tissue cells use the fat as an energy source, sparing the use of glucose, which will then inadvertently increases blood glucose levels. During the postabsorptive state, the sympathetic nervous system is triggered. Adipose tissue is innervated by the sympathetic nervous system, and it can quickly supply glucose if blood glucose levels are low. Low plasma glucose, the fight or flight response, or exercise can trigger fat mobilization and glycogenolysis. This is initiated by the adrenal medulla's epinephrine release. Metabolism and nutrient flow are also influenced by growth hormone, thyroxine, sex hormones, and corticosteroids. Most of these hormones have an indirect effect. Table 24.4 summarizes the hormonal influences on metabolism. Our two big influences are insulin and glucagon. Insulin stimulates glucose and amino acid uptake by cells, glucose catabolism for energy, glycogenesis, protein synthesis, lipogenesis, and fat storage, and it inhibits gluconeogenesis. Glucagon and epinephrine stimulate glycogenolysis, lipolysis, and fat mobilization, and gluconeogenesis. Please review the remainder of the hormones on this chart to understand their part in glucose regulation. Now, let's focus on the metabolic role of the liver. Hepatocytes carry out about 500 metabolic functions. They process nearly every class of nutrient, play a major role in regulating plasma cholesterol, store vitamins and minerals, and metabolize alcohol, drugs, hormones, and bilirubin. Table 24.5 goes through the metabolic functions of liver. In carbohydrate metabolism, the liver is particularly important in maintaining blood glucose homeostasis. It converts galactose and fructose to glucose, stores glucose as glycogen when blood glucose levels are high, and in response to hormonal controls, performs glycogenolysis and releases glucose to the blood, converts amino acids to glycerol and glycerol to glucose when glycogen stores are exhausted and blood glucose levels are failing, and converts glucose to fats for storage. The liver has the major responsibility of fat metabolism in the body. It is the primary site of beta oxidation. It converts acetyl-CoA to ketone bodies for release in tissue cells, stores fat, forms lipoproteins for transport of fatty acids, fats, and cholesterol in the blood, synthesizes cholesterol from acetyl-CoA, and catabolizes cholesterol to bile salts, which are secreted in the bile. Without the liver's metabolism of proteins, severe survival problems would ensue. Many essential clotting proteins would not be made and ammonia would not be disposed of. The liver deaminates amino acids for their conversion to glucose or for ATP synthesis, forms urea for removal of ammonia from the body, forms most plasma proteins, and functions in transamination, the interconversion of non-essential amino acids. The liver also functions in vitamin mineral storage and biotransformation functions. The liver stores 1 to 2 years worth of vitamin A, stores 1 to 4 months worth of vitamins D and B12, and stores iron as ferritin. It also metabolizes alcohol and drugs by inactivating them for excretion by the kidneys. It processes bilirubin, resulting from red blood cell breakdown, and excretes bile pigments. It also metabolizes blood-borne hormones to forms that can be excreted in the urine. Now let's move on to cholesterol metabolism and regulation. Cholesterol is not used as an energy source. It is the structural basis of bile salts, steroid hormones, and vitamin D. It is a main component of plasma membranes. 15% of blood cholesterol is ingested, with the rest made primarily by the liver. Cholesterol is lost from the body when it is catabolized or secreted in bile salts that are lost in the feces. Lipoproteins transport water-insoluble cholesterol and triglycerides through the blood. They also regulate lipid entry and exit at target cells. All contain triglycerides, phospholipids, cholesterol, and protein. There are several types of transport lipoproteins. HDLs are high-density lipoproteins. They contain the highest protein content and transport excess cholesterol from peripheral tissues to the liver to be broken down and secreted in bile. HDLs also provide cholesterol to steroid-producing organs. LDLs are low-density lipoprotein. They have the highest cholesterol content and transport cholesterol to peripheral tissues for membranes, storage, or hormone synthesis. VLDLs are very low-density lipoproteins and are more than half of the triglycerides with low-density proteins. VLDLs transport triglycerides from the liver to peripheral tissues, mostly adipose tissue. Chylomicrons transport absorbed lipids from the GI tract and have the lowest density and consist almost entirely of triglycerides. Figure 24.24 illustrates the approximate composition of lipoproteins that transport lipids in body fluids. From the intestines, chylomicrons are 80 to 95% triglycerides. From the liver, VLDLs are mostly triglycerides, whereas LDLs are mostly cholesterol. LDLs are sometimes nicknamed the lousy lipoproteins because we do not want very much of them if we want to be healthy. HDLs are returned to the liver and are mostly protein. When we look at recommended fat levels, the maximum total cholesterol recommended is 200 milligrams per deciliter. Higher levels of total cholesterol are linked to atherosclerosis. The more important measurement is what form is the cholesterol transported in the blood. HDL is thought to protect us against heart disease and are considered the healthy lipoproteins, H for healthy, H for HDL. They are healthy ones because they take cholesterol out of our blood. We want greater than 60, but not less than 40 HDLs. LDL cholesterol is bad because it deposits cholesterol in the vessels. We would like 100 or less LDLs, and over 130 is not good at all. Blood cholesterol levels are regulated by the liver. The liver produces cholesterol at a basal level regardless of dietary cholesterol intake. Restricting dietary cholesterol does not markedly reduce blood cholesterol levels. The more important amount is the relative amount of saturated and unsaturated fatty acids intake. Saturated fatty acids stimulate liver synthesis of cholesterol and inhibit cholesterol excretion from the body. Unsaturated fatty acids enhance excretion of cholesterol into bile salts. Trans fats can occur when healthy oils are chemically transformed to solids, like margarine. These have a worse effect on cholesterol levels than even saturated fats. Trans fats increase LDLs and reduce HDLs. Unsaturated omega-3 fatty acids found in cold water fish have lower proportions of saturated fats and cholesterol. They make platelets less sticky, helping to prevent spontaneous clotting, have an antiarrhythmic effect on the heart, and can also lower blood pressure. Stress and cigarette smoking have been shown to lower HDL levels. Aerobic exercise and estrogen increase HDL levels and decrease LDL levels. Body shape also plays a role. Apple-shaped people who carry their fat on their upper body have been correlated with high cholesterol and LDL levels, whereas pear-shaped people who carry fat on their hips and thighs are correlated with lower cholesterol and LDL levels. Previously, risk for developing cardiovascular disease was determined with high cholesterol levels and LDL to HDL ratios. This was later found to be unreliable. Now it's believed LDL levels and other risk factors are more accurate. High LDLs are treated with statins, cholesterol-lowering drugs. It's estimated over 100 million Americans take statins.